Lethal alleles occur when one combination of alleles in a cross is lethal. Most of the time, this causes a shift in the expected ratio. For example, I mentioned that monohybrid crosses should have a 3-to-1 ratio most of the time, but sometimes they won't. They'll have a 2-to-1 ratio instead. When that happens, one of the groups is missing because it is dead. Examples of lethal alleles are things like the yellow coat color in mice, white horses, and achondroplasia. For example, in the yellow coat color, if we start with two individuals who have yellow coats, they have to be heterozygous. When we cross these two individuals together, we get half of the individuals having yellow coats, a quarter of them being white, and a quarter of them being dead. Since we can't always score the dead individuals, instead, we'll see the 2-to-1 ratio. This is because 2/3 of the living individuals will be yellow and 1/3 will be white. Whenever you see this group is missing. Sometimes lethals are homozygous recessive, even though the phenotype is dominant. In this example of a recessive allele, this is the dominant coat color, but recessively lethal. This can make it a little confusing. There are lots of traits that behave this way. For example, the Mexican hairless dog also has to be heterozygous all the time. This is because the hairlessness gene that it is using is homozygous lethal. In a cross between two hairless dogs, you'll expect to get 2/3 hairless and 1/3 wild type, with the last group missing. Most traits that behave this way cannot be true breeding as a result, and they have to be actively selected for if you want to keep them in a lineage. But there are other traits that work this way too in the general population. For example, achondroplasia, which is a common form of dwarfism, is an autosomal dominant trait, but it is homozygous lethal. This means that anyone with achondroplasia must be heterozygous for the trait. Other things that can play with what the ratio turns out is things like penetrance and expressivity, which are not the same thing. Penetrance is a measure of how likely you are to display a phenotype if you have a specific genotype. For example, complete penetrance means that every individual with the same genotype are going to have the trait. But if some individuals with the same genotype do not have the trait, this is incomplete penetrance. This means that the genotype increases the likelihood of having the trait, but doesn't guarantee the trait will occur. Expressivity is different. Expressivity determines the variation in the phenotype expressed. In this case, narrow expressivity means that individuals will all have similar phenotypes. But variable expressivity means that individuals will all have a phenotype, but the severity of that phenotype will be different. So penetrance has to deal with whether or not the trait happens, whereas expressivity has to deal with how consistent the phenotype is. An example of this that is both incompletely penetrant and has different expressivity is polydactyly. Polydactyly is a dominant trait. However, individuals that have polydactyly, at least genetically, may or may not have extra fingers or toes. There's variability in the number of limbs that are impacted and the severity of that impact. In fact, some individuals that carry this trait don't have any extra fingers or toes at all. So this is an example of a trait that is dominant, but also has incomplete penetrance in that some individuals are not affected, and variable expressivity, and that which limbs are affected and how affected they are is also variable. This trait is also conserved. Polydactyly exists in other organisms that have limbs as well. For example, it's a common occurrence in cats, chickens, and lizards. Look at that little extra toe. Things like incomplete penetrance and variable expressivity are part of why genetic testing companies are regulated by the FDA. Because having a disease associated mutation is not usually a guarantee of getting that disease. Usually, these companies and other groups will couch this as a genetic predisposition or an increased likelihood. The other issue with looking at traits like this as a guarantee of having something is because many traits are what we call polygenic, or they experience pleiotropy. Polygenic traits occur when multiple genes control the same trait. And pleiotropy happens when one gene has many functions. So can influence many traits. And then, of course, genes sometimes interact with each other. And a common form of that is epistasis. On top of that, we've talked about how the expression of genes sometimes relies on changes in the environment. So there are lots of reasons why. But I want you to understand that these ideas that genes can have both differences in whether or not they appear that have very little to do with their genotype specifically in the form of penetrance, and differences in how they are expressed in the form of expressivity are part of why it's difficult to determine exactly what every gene does and to tie each individual gene to a specific outcome. Unless noted in your midterm, you should assume that every cross is completely penetrant and that there is no variable expressivity. But you should know what these concepts mean. You should be able to keep them in mind if I asked you to explain why something like an individual has a genotype but doesn't have the phenotype could happen. To understand this a